Hello everyone, welcome back. Today I am here to discuss one important topic in plant physiology, that is vernalization. This word has been derived from a Latin word vernalin, which means spring. So this is the basically the cold requirement, or we can say the low temperature requirement of some winter varieties to produce flowers. So the plants which require this low temperature treatments to induce flowers are known as vernalized plants, and this process is known as vernalization. So we'll discuss about this process in details. Let's start. Today's learning objectives are: we'll discuss about general introduction about vernalization, then its historical background. Then we will discuss about the site of vernalization, that which part of the plant received the stimulus of cold treatment or low temperature. Then we will discuss factors affecting vernalization, and finally the mechanism of vernalization. Firstly, what is vernalization? This word vernalization has its origin from Latin word vernalis, which means spring. So there are many plants which do not come to flower. They are unable to form flowers before they experience a cold temperature or low temperature. So this stimulus of low temperature is compulsory to initiate flowering in some plants. And these plants remain vegetative during the warm season and receive low temperature during winters, grow further, and then they bear flowers and fruits. So if they are not provided with this cold temperature, or we can say low temperature requirement, so they will continuously grow as vegetatively or in vegetative stage. In winters, these plants receives that requirement of low temperature, and after that they will bear flowers and fruits. So this requirement of low temperature to initiate the process of flowering is known as vernalization. So that a low temperature treatment required for promoting flowering was termed as vernalization by a Russian physiologist named as Lysenko in 1928. And the condition occurs in various winter varieties of some annual food plants, like we have wheat, barley and rye, and some biennial plants. Biennial plants means those plants which require 2 successive seasons to complete its life cycle. In first season they grow vegetatively, and in second season they will produce flowers and seeds. So ऐसे plants को biennial plants कहा जाता है. For example, we have cabbage, sugar beet, carrot, etc. And there are some perennial plants like we have Chrysanthemum that also require this low temperature treatment to initiate flowering. In this animated picture, we have seed that is slightly germinated seed. So mostly the part of plant that is growing regions, like we have stem tips, root tips, leaf tips, or developing embryo, that part of the plant or the sites of vernalization, they will receive the stimulus of this low temperature. And if this chilling is not present there, so the plant at ordinary temperature will grow as a vegetative plant. They are not able to produce flowers, and these plants are known as non-vernalized plant. If these plants are provided with a temperature that is for a month at 5 degrees Celsius chilled temperature, then planted at the ordinary temperature, they will produce flowers, and these plants are said to be vernalized plants, और इस process को बोलते हैं vernalization. Lysenko, as we have earlier discussed, Russian physiologist, he observed that there are some plants, like we have some annual and biennial plants, which can be made flower in one growing season by providing low temperature treatment to young plants or moistened seeds. So he observed that there are some plants, like we have some annual plants and biennial plants, they can be made flower in one growing season by providing this low temperature treatment to the young plants. So this process, he called this process as vernalization. So the effect that is induced by the low temperature to the plants is known as vernalization. So this chilling treatment is known as vernalization. The process of shortening of a juvenile or vegetative phase and hastening or fastening, or we can say accelerating the flowering by previous cold treatment, is known as vernalization. So this process is basically known as vernalization. Next comes site of vernalization. Which part of the plant received the stimulus of cold treatment or low temperature treatment? So we have firstly the growing region that I have already discussed, shoot apical meristem or root apical meristem. So these are the regions which contain meristematic cells. These are continuously dividing cells. So the stimulus of vernalization is perceived by only the meristematic cells, and these are present in the growing regions, like we have shoot tip, embryo tips, root apex, developing leaves. So जो साइट ऑफ वर्नलाइजेशन है ये हैं वो पोर्शंस प्लांट्स के विच हैव डिवाइडिंग सेल्स लाइक मेरिस्टेमेटिक सेल्स, और इसमें आता है शूट टिप, एम्ब्रियो टिप्स, रूट अपेक्स और डेवलपिंग लीव्स. सो दीज आर द साइट्स ऑफ perception of vernalization. This is the developing embryo. So developing embryo is also a site of which receive the stimulus of vernalization. Then root tips, which contain the root apical meristem. So this apical meristem region also receive the stimulus of this low temperature. Then the leaves, leaf tips. So these are the sites of vernalization. Next, requirements of vernalization. So there are various requirements. Firstly, low temperature requirement. So low temperature requirement for the plants to induce vernalization is usually 0 to 10 degrees Celsius. It can vary from plants to plant, like it is 3 degree to 17 degree in case of biennial plant, that is a long day plant, henbane, Hyoscyamus niger. Effect of vernalization decreases temperature below 0 degree Celsius. So अगर temperature 0 degree से कम हो तो effect इसका reduce हो जाता है, कम हो जाता है. Low temperature treatment should not be immediately followed by a very high temperature, that is about 40 degrees Celsius. Otherwise, the effect of vernalization is reversed or lost. And that process is known as devernalization. And this phenomenon is known as devernalization. Next factor that is required for vernalization is period of low temperature treatment, that is duration of low temperature. It varies from few hours to few days, and it depends upon the it varies from plant species to species. Lang, a scientist who found that all temperatures from 3 degree to 17 degree are effective if a period of vernalization is 105 days. He worked on this long day plant henbane. If vernalization reduced to 15 days, then the temperature of 10 degree Celsius found to be suitable. If this vernalization reduced to 15 days, means reduced by 15 days, then the temperature of 10 degrees found to be suitable to induce the effect of this low temperature. If temperature was extended to 42 days, the most effective temperature were found to be 3 to 6 degree. So these are the various observations of Lang for different temperatures and the requirement of low temperature. In celery, 8 days of cold treatment can induce significant acceleration of flowering. So celery require 8 days of cold treatment that can induce flowering. So this is the photograph which shows flowering in celery plant. Some plants require several months cold treatment, and that is a prolonged period of cold treatment, or we can say low temperature requirement for a longer duration to induce flowering, and this phenomenon is known as over vernalization, और इस process को कहा जाता है over vernalization. Next factor is age of plant. In cereals, vernalization is effective as soon as germinating seeds have received the cold treatment. So cereals में germinating seeds हैं, अगर उन्हें cold treatment मिलती है, so they will show the effect of this vernalization, and the time period for this cold temperature should be sufficient. Plants like we have biennials that must reach a minimum size before they can be vernalized. So they should be grown vegetatively before they receive the stimulus of vernalization. For example, we have henbane, that is a Hyoscyamus niger, will not respond to cold treatment. ये cold treatment को respond नहीं करता before 10 days of age. अगर ये plant 10 days से कम है and does not reach maximum sensitivity until it is 30 days of age. So 10 days में ये sensitive हो जाता है towards vernalization, और जो maximum sensitivity है वो ये plant gain करता है after 30 days of its growth. This is the photograph which shows this henbane plant. Next factor is actively dividing cells that we have already discussed. So vernalization does not occur in dry seeds. The seeds must be germinated so that they can contain an active embryo, and these cells of embryo, which are continuously dividing cells, they will receive the stimulus of vernalization. For this, the seeds are moistened before exposing to the low temperature. So जो seeds हैं उनको पहले moisten करना पड़ता है before they are provided with this low temperature, and after that they are able to receive the stimulus of this low temperature. In whole plants, an active meristem is required, that we have already discussed. Next factor is water. Proper protoplasmic hydration is must for receiving the stimulus of vernalization. जो protoplasm है वो hydrated होना चाहिए so that the plant can perceive the stimulus of vernalization. Then oxygen is also an important requirement for vernalization. Absence of oxygen makes the plant unresponsive to low temperature treatment, even if water is available in the suitable amount or adequate amount, for example in case of handmaiden. So oxygen is also an important factor to perceive the effect of cold treatment. Next, carbohydrates. Seeds of Arabidopsis thaliana are sensitive to vernalization, and it decreasing with development of seedling. So जो vernalization का effect है वो decrease होता है, sensitivity decrease होती है जैसे जैसे plant की development होती है, and it reaches maximum when the seedling is 2 week old. Why this occur? Why this happen? During the germination of seed there is plenty of carbohydrate, and after its germination the source is depleted, और जैसे ही first 2 leaves बनते हैं they start the process of photosynthesis and they form starch. Then after the formation of starch by photosynthesis, this sensitivity also increases. So that's why carbohydrate is also important to receive the stimulus of vernalization. Once again it increases as the new leaves develops, because after the formation of new leaves it starts the process of photosynthesis, जिससे कि carbohydrates की synthesis increase होती है plant में और sensitivity भी increase होती है. This phenomenon can be observed in Paticus winter rye. So this this variety of rye, it also shows sensitivity toward the cold treatment. So the temperature range is on x-axis, and on y-axis it is progress to flowering. As the temperature increasing from -5 to 13 degrees Celsius, in this range, that is -1 to 11 degrees Celsius, it shows, or 9 degrees Celsius, it shows maximum flowering, and after that it decreases as the temperature increases. Next, protein requirement. The formation of new proteins was found after vernalization in wheat plant. That is, the proteins which are found in spring variety of wheat, same proteins are formed in winter wheat after the wheat plants gets vernalized or they get the cold treatment. So ऐसी protein जो spring wheat में present होती हैं, these proteins are also found in the winter wheat after they are provided with the low temperature. So this formation of new proteins is also an important step which initiate the flowering after getting the vernalization treatment or low temperature treatment. Protein pattern resembles that of the spring wheat. इसमें जो protein pattern है after vernalization, that resembles the spring wheat. So these were the factors which affects vernalization, or various factors on which vernalization depends. Next is mechanism of vernalization and induction of flowering. So there are two main theories. One is the phasic theory. So this theory, the basic principle of this theory is that there are two phases in plants life cycle. One is thermophase, which is required for the vegetative growth, and then comes the photophase, which is important for initiating the flowering process. So Lysenko proposed this theory that every plant possesses or passes through two phases of development. The first one is the thermostage or thermophase, in which the plant grows vegetatively, and the second phase is the photophase. After vegetative growth, the plant require a particular period of light, that is photophase or photostage, that initiate the flowering in plant. So first is thermophase. It is the early phase of life which require definite temperature for development, and in this phase plants grow vegetatively. ये जो phase है इसमें एक definite temperature चाहिए होता है plant को growth के लिए, और vegetatively plant grow करता है and prepare itself for the flowering process. Then it is followed by the photophase and the late phase of life, in which plant require definite light requirement for the flowering. This is called photophase, and this phase is known as photophase. So thermophase must be completed before the photophase. So पहले जो plants हैं वो thermophase complete कर लें, means vegetatively grow करें. After that they are followed by this photophase, which initiate the flowering process. So this is phasic theory given by Lysenko. Next comes the hormonal theory. It is proposed by two scientists, that is Grigori and Purvis, 1930. And this theory explains that there is formation of one thermolabile substance known as B from an unknown precursor A, and this B is the substance which stays for some time. ये persist करता है थोड़े time के लिए plants में. जब plant receive करते हैं cold treatment को, then it is slowly converted into another substance, that is C, and after that this C, after the exposure to photo period, it is converted into another substance. In some books it is written as D, and also known as florigen, that is a hypothetical hormone of which induce flowering, and this results in flowering. But when the low temperature treatment is immediately followed by the high temperature, that is approximately 40 degrees Celsius, then the plant will not convert B into C, while the C is converted back into B, and this B will form another substance. In some books it is written as E, and the plant gets devernalized, और जो plant है वो flowering show नहीं करेगा, it will grow vegetatively, और ये plant grow करता है vegetatively, and this process is known as devernalization. इसे कहा जाता है devernalization, और जो plant है वो flowering नहीं show करेगा. So this theory was proposed by Purvis and Grigori in 1930, proposed that plants synthesize a thermolabile substance which is known as B, and it is formed from some unknown precursor A. Then synthesis of B is accelerated by cold treatment. जो B है उसकी formation speed up होती है by cold treatment, and B persists for a period of time. जो B form है वो persist करती है थोड़ा time के लिए once it is synthesized. Then this B is converted into C form and then to D form, which is also known as florigen, is under the control of photoperiods. So जो ये conversion है C to D का, it is under the control of photoperiod. Means it requires exposure to a light duration. Then this substance D, that is florigen, is supposed to be the flowering hormone, and C is an intermediate compound. जो D है that is known as this flowering hormone, और जो C है that is an intermediate. This conversion of B to C is slow. जो conversion है B to C का, it is very slow, but once it is formed, it is rapidly converted into D. So जो B का C के लिए conversion है that is slow, but once the C is formed, then it is immediately converted into D form when it get a proper light duration. Once D accumulates to a critical level, it induces flowering in plants. If vernalized plant is kept under short day condition or is provided with a high temperature, so the conversion of B to C is inhibited. So जो ये conversion है B to C का, ये inhibit हो जाता है. Instead this C is converted back to B. So जो इस process में जब short day condition provide करते हैं या high temperature provide करते हैं, so जो C form that is converted back into B form, and then B is converted into E form, that shows the devernalization process. It means जो plant है वो vegetatively grow करेगा. So thus the plant remains in vegetative condition. Under long day condition, B is slowly converted into C form, and C is rapidly converted into D form, that is the flower hormone, and the plant will produce flower. So critical level of D ensures the flowering. So this is all about the hormonal theory, which is provided by or proposed by Purvis and Grigori. Next, Mikhail Chailakhyan in 1968 suggested that there are two substances which are involved in flower formation in plants. One is gibberellin, that is Ga, and anthycin, a hypothetical substance like vernalin. So basically, in long day plants, this anthycin is present, and after exposure to a long day condition, this anthycin or this vernalin is converted into gibberellin, and that induces flowering in long day plants. While in short day condition or short day plants, this anthycin is absent. Anthycin absent होता है, so there is no conversion of vernalin to gibberellic acid. So जिससे कि जो vernalin है convert नहीं होता है Ga में, और इसकी वजह से जो plants है they don't show flowering, और इसलिए short day plants में flowering नहीं होती है due to lack of this anthycin. Plants that require low temperature or long days or both for flowering might lack sufficient Ga. ऐसे plants जिन्हें या तो लो टेम्परेचर की रिक्वायरमेंट होती है या फिर लॉन्ग डेज चाहिए उन्हें, और ऐसे प्लांट विच रिक्वायर बोथ ऑफ दीज कंडीशंस फॉर फ्लावरिंग, दे माइट लैक सफिशिएंट जिब्रेलिक एसिड और जिब्रेलिन्स अंटिल दे आर एक्सपोज टू लो टेम्परेचर. जब तक कि उन्हें लो टेम्परेचर में एक्सपोज ना किया जाए या फिर लॉन्ग डेज में एक्सपोज किया जाए, आइदर दे रिक्वायर बोथ ऑफ दीज कंडीशंस. सो इन शॉर्ट डे प्लांट्स दे माइट कंटेन सफिशिएंट Ga, they have sufficient Ga but lack anthycin. So this Chailakhyan observed that the vernalization requiring plants produces vernalin. So जो भी plants जिनको low temperature चाहिए, they produces a hypothetical hormone that is vernalin when subjected to low temperature, and then vernalin is converted into Ga. उसके बाद जो vernalin है वो convert होता है Ga में, that induces the flowering, और ये जो conversion है it is induced by the exposure to light duration or long days that accelerate the conversion of this vernalin into GA. While in case of short day plants, they lack the xanthocin, so there is inhibition in conversion of this vernalin into GA that results in no flowering in plants. Next, there were another scientists like Lang and Melchers. They demonstrated that in long day plant, that is henbane, Hyacinthus niger, translocation of vernalization stimulus takes place through a graft union. So जो वर्नलाइज्ड प्लांट का पोर्शन है, अगर उसको हम ग्राफ्ट करते हैं अनवर्नलाइज्ड और डीवर्नलाइज्ड प्लांट पे, सो वो प्लांट का पार्ट भी फ्लावरिंग इंड्यूस करेगा. दैट प्लांट विल आल्सो शो फ्लावरिंग. For example, this is a vernalized plant. If it is grafted with the plant which is not vernalized, so जब इन्हें ग्राफ्ट करेंगे, so this stimulus of vernalization gets translocated through this graft union to the unvernalized plant, and it will induce flowering in their particles also. और जो unvernalized plant है, that will not show flowering. So if leaf or stem of a vernalized plant is grafted on an unvernalized henbane plant, the latter plant will flower. So जो unvernalized plant है उसमें भी flowering होगी. So it means जो stimulus है vernalization का, that gets transferred from that vernalized portion to the unvernalized portion through that graft union. The stimulus was found to be non-specific. It means it is not a part specific, that is, it can pass across a graft union between the plants of different species. It was Melchers who, for the first time, suggested that a substance, which he called vernalin, was produced during the process of vernalization. So he was the first person to use the term vernalin for that hypothetical hormone. Attempts to isolate and chemically identify vernalin have not succeeded as yet. Next comes chemical nature of vernalin. This is a hypothetical hormone and cannot be isolated. Plants requiring vernalization contain one important thing, that is flowering locus C protein, and this protein is bounded to a specific location on chromatin or DNA that represses genes required for flowering. So this protein, that is FLC protein, it inhibits the flowering process because it is bounded on the portion of chromatin that contains the genes which are required for flowering and keeps in repressed it till the cold treatment is given to the plant. So during cold treatment, this the FLC, that is flowering locus C protein, that dissociates from the loci and allow other factors to bind and activates the genes which expresses itself when this FLC gets dissociated after getting the cold treatment. So जो FLC protein है, ये तभी dissociate होता है इस portion से जहाँ पे flowering genes होती हैं when the plant gets the cold treatment. And after that, the genes gets activated and they will express and induce flowering in the plant. So FLC has a key role in initiation of flowering in Arabidopsis. The FLC है flowering locus C, it plays very important role in flowering in case of Arabidopsis. For example, this is a plant of Arabidopsis which doesn't show flowering because it contains this FLC protein that is binded over the genes which are required for flowering. Once the plant gets cold treatment, this FLC gets dissociated from this chromatin, or the genes हैं, they gets exposed, or they will bind with other activating factors, and under the influence of those activating factor, it will undergo the process of transcription, translation, and express itself, and as a result of that, plant produces flowers. So this is very important protein, that is FLC, flowering locus C protein, which is responsible for repression of flowering until the plant is exposed to the cold temperature. Next comes genetics of vernalization. There are three genes of vernalization, that is 1, 2, and 3, or abbreviated as VRN 1, 2, and 3. These three genes plays key role in deciding the role of vernalization in cereal grains. All three genes have now been isolated. These three genes have been isolated. VRN1 encodes for a transcription factor that induces and induced by vernalization treatment. So जो VRN1 है, ये gene encode करती है transcription factor को, which activates the transcription process, and this is induced by vernalization treatment, that is cold treatment, low temperature treatment. Then there is another gene, that is VRN2, that represses flowering under the long day condition by blocking expression of this floral stimulus FT. So जो VRN2 है, ये जो gene है, ये ब्लॉक करती है फ्लावरिंग के प्रोसेस को लॉन्ग डेज में बाय ब्लॉकिंग एक्सप्रेशन ऑफ द फ्लोरल स्टिमुलस FT. then VRN2 itself is replaced by and repressed by VRN1. so जो VRN2 जीन है, ये रिप्रेस होती है by VRN1. Then VRN3, it is the in cereals, it is equivalent to the flowering locus T, that is FT in Arabidopsis. जो फ्लावरिंग लोकस टी है अरबिडोप्सिस में वैसे ही काम करता है ये VRN3 और ये रिप्रेस करता है फ्लावरिंग को. The next comes devernalization. Vernalization can be reversed if immediately followed by a high temperature treatment, and such reverse effect is known as devernalization. जब हम प्लांट्स को एक्सपोज करते हैं लो टेम्परेचर में, then that is immediately followed by high temperature, that is approximately 40 degrees Celsius or above 40 degrees Celsius, then the plant shows a reverse effect of vernalization. That reversion of vernalization is known as devernalization. Most plants have neutral temperature where neither vernalization nor devernalization occurs. For example, 15 degrees Celsius for Peticus rye. So there are plants which have the range of temperature जिसमें ना ही vernalization का effect show होता है और ना ही devernalization का process show होता है. That temperature is known as neutral temperature. For example, we have Peticus rye जिसका neutral temperature है 15 degrees Celsius. This results in the delay of flowering phase in plants. और इसके रिजल्ट से जो फ्लावरिंग फेज है वो डिले हो जाता है. इट मीन्स द प्लांट्स continues to grow vegetatively. So in this picture we can see here, during vernalization process there is compactness of these chromatin fibers, while under devernalization condition जो ये chromatin fibers हैं और nucleosomes हैं वो relaxed होते हैं. Under vernalization condition they gets compressed and they will show the effect. There are some genes which gets expressed under vernalization condition. Next comes importance of vernalization. So जो vernalization का concept it can be used in agriculture and horticulture. Vernalization can help in shortening the juvenile or vegetative stage and produce early flowering. Plants में हम इससे early flowering produce कर सकते हैं, means flowers induce कर सकते हैं by providing low temperature. It is applicable not only to temperate plants but it can also be applied for tropical plants. For example, we have wheat, rice and millets or cotton. Then it increases yield. ये जो production है उसको increase करता है, and it also develops resistance against many diseases and cold. Then it removes kernel wrinkles. जो seeds हैं उनके wrinkles को remove करता है in case of critical. It also enables biennials to behave as annuals. जो biennial plants हैं अगर उनको vernalize किया जाए, so they can behave as annual plants. Plants can be grown in such regions where they normally do not grow after providing them low temperature. This was all about vernalization concept and various topics related to vernalization. Hope you will get some idea from this presentation. If you have any questions, queries and any suggestions, you can give it in comment section. Thanks for watching. Have a great day.